Hi, we're continuing with cell biology and life science in a nutshell. So we're learning about cell division. So there's we're going over mitosis. This is prophase stage. Um, this picture is showing dividing cells, and their blue is the nuclear envelope of the nucleus. With condensed chromosomes in red, when the chromosomes double themselves. So I guess this is before prophase, the first stage of the cell division, which we just discussed. So the DNA replicates, all the mass of the cell in general increases, the protein's content increases, so whatever molecules are needed are getting ready, and for cell division to occur, the mitotic spindle is set up, and yeah. So the nucleolus, which is part of inside the nucleus, disappears. So the normal functions of the cell stop for the cell division to occur. The chromatin, which is the genetic material in the they are loose in interphase, which is before cell division. Then they become more condensed into chromosomes when cell division begins, so they can move around and more easily split themselves into two separate cells. So then you have sister chromatids, which are identical copies of each chromosome joined by a DNA element called the centromere, which is a protein unit. Okay. So the cytoskeleton is helpful with the movement of the DNA and the chromosomes. In metaphase, the spindle fiber spreads out throughout the cell, bringing the chromosomes and chromatids to the center of the cell. Chromosomes are at their second most condensed and coiled stage. These chromosomes carrying genetic information align in the equator of the cell before being separated into each of the two daughter cells. Microtubules formed in prophase have already found and attached themselves to kinetochores in metaphase. So the kinetochores are the units on the chromosomes themselves, so that bind to the rest of the cytoskeletal spindle fiber. System, the fibers. Then in anaphase, the cell starts splitting into two cells as the chromatids pull apart. Replicated chromosomes are split. The daughter chromosomes are moved to opposite poles of the cell. Chromosomes also reach their overall maximum condensation in late anaphase to help chromosome segregation and the reformation of the nucleus. Securin is a protein which inhibits a protease known as separase. The destruction of securin unleashes separase, which then breaks down cohesin, a protein responsible for holding sister chromatids together. The centromeres are split. The new daughter chromosomes are pulled towards the poles, as shown in this picture. Then in telophase, which is the final stage of division, further spreading of the spindle fiber brings genetic material into two separate nuclei. However, the cytoplasm is still shared. Reappearance of the nuclear membrane and nucleolus occur. Two daughter nuclei form in each daughter cell. The phosphatases dephosphorylate the nuclear lamins at the ends of the cell, forming nuclear envelopes around each nucleus from components of the parent cell's nuclear envelope. So that's how the envelope reforms by dephosphorylation. And the chromosomes reach the poles. The remaining spindle microtubules begin to depolymerize, so come apart. The chromosomes also unwind back into their uncondensed or expanded chromatin in their new nuclei. Lastly, you go back into interphase. The closed cells result as a result of cytokinesis, or the splitting of the cytoplasm. Each cell has its own nucleus genetic material inside. And the chromatin are not condensed any longer. In interphase, this is when the cell undergoes its regular normal functions. Interphase is where the typical cell spends most or 90% of its lifespan. The daily metabolic phase of the cell. It obtains nutrients, metabolizes nutrients, grows. It reads its own DNA, producing proteins, etc. During this phase, the cell copies DNA. When it gets ready to do mitosis or cell division, or meiosis, depending on if it's a regular somatic body cell or if it's a reproductive cell. So again, somatic cells are normal diploid cells of the body. They go through mitosis in order to reproduce themselves. Whereas diploid germ cells are like the cells of the reproductive system that undergo meiosis. These are called primary spermatocytes in males and oocytes in females. So they go through meiosis to create haploid gametes. The gametes are called sperm and ova for the purpose of the sexual reproduction. So haploid gametes combine and recombine their genetic material to form the offspring cell or the zygote. And in meiosis, the stages are pretty much similar to mitosis, and we went through them in earlier reading. So then, before you have division, again, you have gap 1 phase where the cell grows, and the contents within it double. When you're gonna have cell division occur, and then S phase of right before division occurs, where the synthesis of DNA leads to doubling of genetic material. G2 also, the cell contents are growing, and so then interphase leads to prophase, and you just have that cycle of prophase to metaphase to anaphase to telophase to interphase. We'll continue in the next part. Thank you.